So this series of lectures is on Chapter 13, Microbe-Human Interactions, Health and Disease. My lectures start with the material from the end of Chapter 13, which is a focus on disease and epidemiology. Then in the next series of lectures, we'll return to the beginning for mechanisms of pathogenicity. Epidemiology is the study of how diseases spread through a population. So first we'll focus on how pathogens enter a human host, how they exit, how they spread. After that we'll focus on exactly how they create disease once they're inside the human body for the mechanisms of pathogenicity part. And just FYI, I am making these recordings in my home with the whole family present because we're all staying in for the coronavirus. So I apologize if there's a lot of background noise, children in and out, things like that. Just so you know, the situation in advance is kind of ironic, isn't it? Or maybe it's just a very appropriate time for this lecture. All right, so for this lecture, we're going to cover material that's important to understand patterns of disease spread in a population. So like reservoirs, where do diseases come from? Are they communicable or not? How are communicable diseases spread between hosts? And then in the next lecture, we'll cover classification of infectious diseases, and then after that, nosocomial infections and emerging infectious diseases addendum. I know we already covered EIDs earlier in the term, but this addendum will be useful now, after we talk about diseases spreading. Right. So a disease reservoir is an animal or an environment where the disease is commonly found. Sort of like the source of the disease from which it spreads. So a human reservoir is pretty self-explanatory. This is when the disease lives all or most of its life cycle in the human body. That's when humans would be the reservoir of the disease. So some examples would be HIV, typhoid, streptococcal, also the novel coronavirus, COVID-19. Those are all examples of a human reservoir. Diseases that complete most of the life cycle in other animals, or which more commonly affect another species of animal but can be transmitted to humans, those we call zoonoses. So examples would be rabies, Lyme's disease, West Nile virus, bird flu. Those are all diseases or pathogens that have an animal reservoir. So we call them a zoonoses. And then non-living reservoirs. So these are diseases that spend most of their life cycle in the environment, not infecting anybody. So soil and water, those would be the most common environmental sources of a pathogen. Often these are accidental pathogens. They're not truly designed. These are microbes that are not truly designed to spread through living hosts. And so infecting a human is kind of a dead end for them. So I like to call them accidental pathogens. Sometimes human pathogens can be transmitted through something like water, and so water may be a reservoir for a human disease. So these reservoirs can be sort of confusing because when we're talking about something like cholera that spends a lot of time in people but also a lot of time in the water, we think, is it a human reservoir or a non-living reservoir? And typically the literature would say that that's a water reservoir because that's where a human would get it more directly. So it's alternating in its life cycle between human water, human water, human water, malaria, human mosquito, human mosquito, human mosquito. So in the case of malaria, we might say that it has an animal reservoir, even though it's alternating back and forth. In cholera, we'd say it has a water reservoir, even though it is alternating back and forth. Diseases like tetanus and valley fever, we'll come back to those, but those are more like accidental pathogens. So they don't have a way of exiting their host once they're in. Being in a host is a dead end for them. We find them in the environment, but they're not true pathogens. They're not evolved to be pathogenic or to spread through a population. So that brings us to non-communicable diseases. So non-communicable diseases are diseases that cannot be spread from one person to another. So in some cases, these are going to be diseases that are caused by normal flora organisms. So what I mean by that are common microbes found in everybody's microbiome. You can't transmit them to another person because the other person already has these microbes. So these are diseases from within. The reason you would get a disease from a normal member of your microbiome is because maybe that member of the microbiome got into an area of the body it's not supposed to be. So one example would be E. coli for urinary tract infections. Everybody has E. coli in their gut. If your E. coli gets into your urinary tract, it can cause an infection. So you're not going to transmit that to another person because everybody has E. coli. So that's a non-communicable disease. Another example would be if somebody has some problem with their immune system, they're sick with something else, they have an autoimmune disorder, or some dysbiosis in their microbiome, so microbes in their normal microbes are out of balance. So one example would be Candida for yeast infections. So everybody has Candida living on their skin, on their genitalia, but if for some reason there's an imbalance and it overgrows, and that can have to do with the host's health. So again, you can't transmit that. Everybody already has that. There's something going on with the health of the host that's making their normal microbiome become a little bit disrupted. And another example that I have on the slide, heart infection with mouth bacteria after dental surgery. So, one rare complication of having major dental surgery is that the bacteria that's normally found in the mouth gets into the bloodstream and moves to another part of the body, like the heart. So this is one of the reasons it's important to take antibiotics after any kind of surgery. But that's another example of bacteria that's normal and benign in one part of the body can cause disease if they get to the wrong part of the body. So another sort of non-communicable disease can be from food poisoning due to preformed toxins. So the two examples I have up here are staphylococcal food poisoning and botulism in adults. So in both of these cases, the microbe can get into the food, and the microbe can, while it's living in the food, it's metabolizing, it's breaking down the food and making waste products, and the waste products are pathogenic to a human who might ingest them. So in the case of staphylococcal food poisoning, the staph can contaminate the food, but the staph is destroyed or killed at cooking temperature. So you cook the food, the staph is killed, but for some species, the toxins they leave behind are not broken down at cooking temperatures. And in that case, the food could be cooked. The person ingesting it would still get this stomach upset from the toxins left behind by the staphylococcus. Botulism is the same idea. So botulism, Clostridium botulinum, that's an anaerobic bacteria. If this bacteria gets into food that's then canned and put into an anaerobic environment, remember, we've talked about Clostridium before, they form endospores. So the Clostridium will be in an endospore form once exposed to oxygen. Then once in the can, it will germinate because that's its preferred environment, in an anaerobic environment. And while it's living in that can of food, it will multiply and it will metabolize and it'll excrete this toxin to help it metabolize its food. Then you open the can, it's exposed to oxygen and it dies. You cook it also and it dies, but the toxin there could still be some toxin left behind. In that case, botulism toxin can be broken down at boiling point. So those are cases where there's no bacteria infecting the human host. So there's no bacteria actually causing disease. It's a toxin left behind from a bacteria that's now gone. And so this is more like poisoning. So if you take a poison, you can't then communicate that poison to somebody else, right? It's not contagious because there's no bacterial component. And then a final example of non-communicable diseases are diseases due to organisms normally found in the environment but who are accidental pathogens. So some diseases from the environment are communicable, but some are not. So again, the non-communicable diseases from the environment are accidental pathogens. Infecting a person is not useful to them. They have not evolved to spread through a human or an animal population. The animal body is a dead end for them. So they can enter accidentally, but they have no exit. They have no way to move on to the next person. And so the reason these kinds of microbes might be disease-causing is sort of accidental. Their toxins are designed to help them to survive and compete in their natural habitat, in the soil, for example, and those toxins just accidentally cause problems for animal biochemistry. So tetanus and valley fever are examples. Tetanus, the disease is described by paralysis at its most extreme. Valley fever by chest pain, cough fever. So, for example, tetanus, the toxin that creates paralysis in a human is actually designed to help that bacteria break down amino acids. So tetanus is just trying to break down amino acids in its natural soil environment. Once it starts producing those enzymes in the human body, it disrupts the neuromuscular system. So we tend to worry more about communicable infections because they spread through the human population, and a lot of people can then, one person can then infect many people. There's two main ways a communicable disease can be passed from person to person. One is vertical transmission and the other is horizontal. So vertical transmission is really, really specific. That's from mother to child. So that can be transplacental or during labor or through breast milk. So any of those routes, transplacental during pregnancy, through the blood during birth, or passed through breast milk. So that's specifically mother to child. Then horizontal communication of disease can include many things. Saliva, skin-to-skin contact, fomite transmission. Fomites are inanimate objects. The fecal-oral route, vectors. These are usually, insects that bite. And other ways, zoonoses, venereal diseases. So we'll cover all of these and how they move from person to person. So let's cover contact transmission first. So again, this is horizontal transmission for communicable diseases. So contact transmission means you need close contact with the person who has the disease in order to pick it up. And there are three subcategories of contact transmission. So first there's direct contact transmission. That's skin-to-skin contact or direct transfer of body fluids. So some examples would be human papillomavirus, which can cause cervical cancer, MRSA, which stands for methicillin-resistant Staphylococcus aureus, human immunodeficiency virus or HIV. So all of those require really close contact, so again, skin-to-skin or exchange of body fluids. The second kind of contact transmission is indirect contact, and that's through fomites. So fomites are inanimate objects on which a disease might survive for a short period of time, long enough to get from one host to the next. So some examples of fomites could be diapers, towels, tissues, toys, money. Needles are actually considered to be fomites, so that can be a little bit confusing because blood-borne diseases can actually be transmitted through fomites through a needle. So some examples of diseases that are spread this way are hepatitis B, tetanus, the flu, pinworms. Those can all live for at least a period of time on an inanimate object and be transferred from one host to another through this object. The third type of contact transmission is droplet transmission. This is different than airborne transmission. So droplet transmission refers to diseases that can only be spread through heavier, larger droplets, like in a sneeze. So diseases that can spread really long distances or stay suspended in the air, they're still suspended in droplets, but these droplets are so small we say that they're aerosolized. And we don't consider that contact transmission. So droplet transmission and airborne transmission are very similar, but we classify droplet transmission as a style of contact transmission because you have to be really close to the infected person in order to pick up the disease. So this requires larger mucus droplets, which can't travel too far, usually less than a meter from the person spraying them, from the person sneezing or coughing or speaking. So some examples of diseases that are spread through droplet transmission are influenza, pneumonia, and also the novel coronavirus. Vehicle transmission. So we just did contact transmission, and we did 3 styles of contact transmission. So next we're going to work on vehicle transmission. And there are 3 styles of vehicle transmission as well. So we have waterborne transmission. This is when, so a vehicle, think of a vehicle as just like something that the disease is riding on to get farther distances away from one person to the next. So first style will be waterborne transmission. This is when the disease travels between hosts in the water, so like cholera or giardia. Foodborne transmission, this is when the disease or the pathogen is traveling between hosts in food. So if the food is undercooked, if it was prepared in unsanitary conditions, if it wasn't refrigerated well, those are the kinds of ways a pathogen might live in food to get into their next host. So some examples would be tapeworms or food poisoning. There's a lot of different bacteria that can cause food poisoning, but they travel through undercooked or poorly prepared food. And then the third style of vehicle transmission is airborne transmission. So this is similar to droplet transmission, but you don't have to be near the infected person to pick it up in this case. So this is when the droplets are very, very fine, so fine spray that can travel far, much farther than a meter. Some examples would be tuberculosis or pertussis. So tuberculosis, for example, can stay suspended in a room for extended periods of time. So a person could be in a room, cough, leave the room, another person could come in, breathe in the air, and become infected with tuberculosis. That's very different than droplet transmission, where you really need to be near the person who's infected in order to come down with the disease. So tuberculosis, pertussis, these are so contagious because they are airborne. So we had contact transmission and 3 styles of contact. We had vehicle transmission, 3 styles of vehicles. And now we have vector transmission. There will be 2 styles of vector transmission. So vectors are animals that act as a vehicle. So you can, it's sort of a gray area in a way between vehicle and vector. You can just think of a vector as a living vehicle. So these are animals that are moving the disease far, allowing the pathogen to travel very far from the original host to the next host. So the first style of vector transmission is mechanical transmission. And this is when the pathogen is acting as a passive passenger on an animal body part. So for example, usually the example is a fly lands in maybe feces that's infected with some pathogen, then and picks it up on its legs, picks up the pathogen on its legs, and then lands on your food or something. So the pathogen is not actively using the fly to get around. It's just, it's more just, again, a passive passenger on the fly. So some examples of pathogens that can be moved around this way are typhoid fever, bacillary dysentery, things like that. But usually when we talk about vector transmission, we're talking about biological transmission. And this is when a pathogen is actively transferred through a bite or through vector vomit, feces. So often a biological vector is, the pathogen is actively completing part of its life cycle inside that other animal. So some examples would be Lyme's disease. So Lyme's disease lives in the tick for a period of time and then is transmitted to an animal host through a bite, right? Rabies, some other parasites, malaria would be a good example as well. So with malaria, you have the pathogen actively completing part of its life cycle in the mosquito and then requiring another animal host to finish its life cycle, like a human host to finish its life cycle. All right. So in chapter 13, when we're talking about how diseases spread, I want you to remember these 5 steps for how a disease moves through an individual human host. So there's going to be a portal of entry by which the pathogen enters the host. Then the pathogen has to adhere to host tissue and host cells. Third, the pathogen has to evade or penetrate the host's defenses. The host defenses are going to include various aspects of the immune system. Fourth, the pathogen is going to cause damage to the host cells. If it didn't damage the host cells, it would not be considered a pathogen. 5, the pathogen has to exit the host. So for right now, we're talking about epidemiology and how pathogens spread through a population. So we're going to focus on portals of entry and portals of exit. And then in the next series of lectures, when we cover microbial mechanisms of pathogenicity, then we'll talk about what's happening in those 3 steps in between, what's happening while it's inside the human body to cause disease. So remember, like all living things, the goal of the pathogen is to replicate. Pathogens that replicate successfully are the ones that persist. So obviously if it does not replicate well, the pathogen lineage is going to end. Human diseases are microbes that have to take advantage of a human body in order to replicate. So that's why they have to enter and cause damage. So what they do inside the human body is they're taking advantage of your body's resources. So they're using your body for food, they're taking your nutrients, they're spreading through your body, so they're breaking down barriers, breaking down connective tissue. Those are the kinds of things that make you sick. And then they have to find a portal of exit because your immune system is going to catch on eventually, either that or they're going to destroy you. So there's only, for a pathogen, there's only two ways this can end. The immune system kills the pathogen or the pathogen kills the human host. So they have to find a portal of exit. And that's different from the microbes that are part of your microbiome. Those that are living peacefully with you, they're not in a hurry to find an exit, right? Because they're living more sustainably with their host.